P.Z. Myers, a professor of developmental biology at University of Minnesota Morris. Yes?
Right.
All right. I have invited you on here again to because you are a developmental biologist, and occasionally I get comments from people who don't really like reality a whole lot and want to come up with an alternative. And in this case, I've heard this a few times. I don't know where it originated from, but there's this issue with protein families, not just protein folding, which used to be an argument I used to hear a lot, but this one is with protein families. And that the person, the people that bring this up are all over the place as to what they're talking about. So it was a little difficult having an interaction with this guy back and forth because I knew as soon as he started bringing it up that I'm going to talk to PZ about this, but I couldn't get the guy nailed down as to what the hell he thought he was talking about. So he said there is no research, and this is interesting. There is no research that says that protein evolution is possible unless you appeal to evolution. That's circuit. By the way, he has absolutely no idea what evolution means. He keeps arguing that he accepts that all dogs are related, domestic dogs are related to each other, that different species of dogs are related to each other and to foxes as well. He's okay with all of Canidae having a biologic relationship. He does not understand that that is evolution. He wants to call it adaptation or anything other than what it is. But to let him continue, there is no research to publish that explains how new protein family with stable novel folds can evolve in the rugged evolutionary landscape. Only conjecture and always with an appeal to the theory. Now, let's just start with that. I remember reading about abiotic proteins being developed in some experiments. Am I wrong in this?
No, the guy is nuts. That's all I can say. Yeah, I mean, because his whole argument is bogus. He doesn't understand where the theory is because he's arguing, okay, you got to appeal to evolution as if the appeal to evolution is the evidence, and it's not.
I
What
tried to
we
explain
have,
to him that evolution is descent with inherent modification, that it is a change in allele frequencies over reproductive populations over many generations, and that the way to see evolution is when you have one original ancestral group that produces a variety of descendant subsets at the population level. That is evolution. So if you accept that breeding that new variants or subspecies or subbreeds of dachshunds, even any of that, it's all evolution. And he just wouldn't accept that. He criticized me for being stupid and not knowing that that's just adaptation. And then I had to show him the definition from Evolution 101 on UC Berkeley that talks about how adaptation is defined as evolution under the influence of natural selection.
Yeah, well, this is a story of molecular evolution, and where he's getting confused or is intentionally being obtuse is that what we do is we go in and we observe the genome. We look at the structure of the genes. We look at the sequence, the genetic sequence of these things. And we see that, for instance, there are these gene families, like the globin gene family or like the transporters, various transporter gene families. And we look at individual members and compare them to other members, and they're remarkably similar. They show amazing similarities between each other. And so then what we have to do is explain that, right? That's where we deploy the theory of evolution. We say, okay, we got all these observations. Here's all this data. Here's a good explanation for all of this similarities. But the theory is not the evidence. It's never the evidence. The evidence is
What
the
he's
evidence.
asking for, I
Yeah.
guess, is if you're saying that we theorize that we have these that this is a likely explanation. Is that what he's saying? That by the circular reasoning back to the theory. So he's wanting us to show that novel proteins can come about through an evolutionary process.
Well, but here's the thing. We don't need an evolutionary process for that.
Okay.
We're not looking. We don't have to be looking at different organisms and different lineages to put this together. Although we can integrate everything into an evolutionary framework. But what he's arguing is that we don't see any evidence for how these could be assembled. When you get into the literature and you look in the scientific literature on, for instance, transport protein evolution,
Okay.
what you discover is that not only are there these deep similarities between different transport proteins, but when you look within a single transport protein, there's clear evidence of intragenic duplication to build that structure. So, what we're doing is we're just doing simple genetics and molecular biology, and those highlight various similarities. And there's no mechanistic obstacle to this happening. I mean, gene duplication and intragenic duplication are known common phenomena. So we got all that there. And then we have to explain, well, how did all these arise? Well, it's theory of evolution we come back to explain it. This guy does not offer an alternative, I notice. He simply
Yeah,
just
he says he
denied.
doesn't have to. That as long
Yes,
as this is impossible, that means that creation is possible. That's honestly his logic. And
but
I
it's not
well,
impossible is the thing I'm saying is, yeah, we can break it apart piece by piece and we can see how the changes occurred to assemble these proteins. And there's nothing remarkable about it. It happens all the time. And we can see how these different components built these different proteins, and then how the different proteins were duplicated. For instance, you look at the globin gene family. There's two globin gene families, but each one of those is in this tightly packed little region, and we can actually look at that whole region and see the hallmarks of gene duplication that produced globin alpha and beta and gamma and all those things in that region. So there's absolutely no molecular or genetic obstacle to these things happening. He just wants to say that there is, so
well,
that he
the
can pull back.
interesting thing was what I tried to explain to him over and over again that if we're looking at allelic variants, we're looking at phenotype and genotype where you have two different subsets, both from an ancestral group. That ancestral group produced two or more different variants. They are genetically distinct, where every member of this group has these traits that are not shared in common with any member of this other group, or maybe not with the ancestral group either. That is evolution by definition. He doesn't understand that. He says that he has a problem with gene duplication, gene transfer, gene shift, whatever. I don't.
Whatever
And,
that
and
is.
anything else he may have missed under the influence of natural selection. He says it is insufficient to produce biodiversity. He accepts that it happens, and we have all this genetic proof that it happens, but he's still not going to call it what it is. He still says it's circular to say that evolution is evolution.
Well, to say that is circular, evolution is evolution, circular. Okay, but that's not what's being said here. It's said that these particular specific bits of data, these sequences that we find in the genome,
All
have
right,
to
let
be
me go over
explained.
this. Let me go over this sentence by sentence, and I want to see how you respond to this. There is a barrier to protein evolution, he says. Is there?
No, I mean there's okay. To be thorough, there are functional barriers. So if you can trash a protein with a major mutation, like a deletion or something, so it's no longer functional, and then it can drift out of existence or be selected against. But materially, looking at the genome, no, you can scramble things a lot and still get functional and interesting proteins.
I hope people don't misunderstand that, because I realize that you teach a classroom full of people who are already partially educated, and I'm dealing with people who are not at all, and who
Which is
often
obvious.
mutations scramble the DNA. That's what I hear. So if you use the word scramble, then people are thinking that you put your DNA under a blender.
Oh, well, no, that's there are various kinds of mutations. I mean, of course, you know all this. There's point mutations,
I have to
there's
do. It
deletions, there's
doesn't matter what I
insertion.
know. I'm trying to reach an audience that doesn't
Right.
know what I know.
So there's various categories of mutations, and most of them do not totally mess things up. What they do is they insert or remove or change little bits and pieces of the sequence. And that's a common phenomenon. That's simple mutation going on. Um, but what we see is that when we look at these various genes, we can see these deep similarities between multiple sequences that are functional but are different. And the best explanation for that is gene duplication and then drift, variation in the genes. For example, I do a lot of neuroscience stuff, and if you look in the neuronal the neuronal toolbox, we have all these ion channels, and you look at the individual ion channels, and it's like unless you look closely, they look the same. They all have these four modules that cross the membrane, transmembrane regions. Uh, and if you look at, for instance, sodium and potassium and calcium ions, ion channels, they look on a superficial level very different or very similar. But when you look at the functional level, they have these differences. You know, calcium channels let calcium through. They don't let sodium and potassium through. And those all those require is small changes in single amino acids to change the diameter of the pore and things like that. Um, and that produces this diversity. So when we look at these channels, we can see that, yeah, this is a big family of structurally similar genes with slight differences between each that have functional significance.
I'm gonna, as I said, I would like to read through this guy's every sentence just to get the your take on it. And his argument actually gets worse as we
Yeah,
go along. So we're going to start with the best that he's got, and then it'll get dumber. So he says that gradual change does not provide a path from one protein family to another because the landscape is rugged. Uh, rugged?
yeah. No, I've rugged landscapes is that's a common term in evolutionary biology. That uh, there are problems with modeling evolution as traversing a landscape. So let's just set that aside for a moment, though. But there are differences between a landscape that is smooth and gradual, and you can easily see how, you know, as Dawkins would say, you're climbing Mount Improbable by moving up it. And then rugged landscapes are, you see a lot of variation. You know, simple sequence changes can cause radical changes in the behavior of the protein. And as he talks about, yeah, it's difficult to climb these hills, and things can get stuck on plateaus or peaks. This is all part of standard evolutionary theory. We know, but that's again not a barrier. That would not a barrier to the general property of evolving proteins. It does mean that some protein sequences might be very difficult to achieve, but
I tried to ask him where, because he said that there can't be any evolution because of the impossibility. He said, where he said not there being no possibility of producing novel protein families. So I had to ask him, where are these protein families? If I showed him a genetic chart for, the evolution of canids, for example, I said, how many different protein families are depicted here? And he said that somehow his concept of protein families prohibits evolution, seemingly at every level, but doesn't show up on any cladogram at any level. But then later, he said that it occurs in the Cambrian explosion where everything appeared rapidly, which of course we know that it didn't. And he said that
yeah,
there were no fossil precursors, which we know better than that too. But he's basing everything on a false premise. And then he says that it was at the origin of superfamilies. So when I brought up to him superfamilies, So between hominoidea and cercopithecoidea, are there different protein families there? And again, there's no answer. We can't get him to pin down where this should be a problem for evolution. He's thinking that if it's at the dawn of eukaryotes, or if it's in the Cambrian, or if there's any chink that he can, if there's any weakness in the armor, then the whole thing will come down. The whole house of cards, in his perspective, doesn't
Yeah.
understand. He's already admitted to evolution without conceding that he admitted that. He unwittingly admitted that, and he unwillingly admitted that,
Right.
which is weird. I
Yeah.
can't get him to understand that. He
Well,
says point mutations will lead proteins off the functional cliff. He says duplication doesn't fit the bill either because there's not enough variety. I don't know what he's talking about because I can't get him pinned down on anything that it would have an impact on what he's talking about.
right. Well, I can sort of vaguely see what he's getting at, and that is that there are gene families that are distinct from other gene families. And I think somewhere in his long text he mentions proteases versus transport proteins.
I didn't
And
see that.
and the idea is, yeah, I think it's near the end of what you sent to me.
Okay.
Let's see, what did he say? He says, I'm talking about entirely new families. Think superfamily, like a transporter to the first protease.
Okay, yeah.
Yeah, so that's what he's arguing. And that's also nonsense because a lot of these super families that he's talking about did arise well before the Cambrian. They were rising in bacteria, and we've just sort of inherited them. I can't
So he's
think.
not talking about a taxonomic superfamily.
No.
Okay. That makes
He's
sense because he told me he wasn't talking about taxonomy, but he says that no evolution is possible. Like, okay, well, you accept different species of dogs. And he said he has no argument with speciation. Well, how is it then that there's no evolution possible?
Yeah. Well, in the case of the superfamilies he's talking about, though, there is evidence of how you could get a new, way back in the distant past, how you could generate a new superfamily, just like, there was an event in the Precambrian that separated various eukaryotes into uh, deuterostomes and protostomes, for instance. Okay, that's a big difference. And this is a difference that we're also seeing in these gene families. And like he says, transporter to protease that's a huge jump. However, it's not an unleapable gap between these things.
He says
That
there has to be a bridge.
Yes. And when we look at these sequences, we can find the bridges in there. So as I mentioned earlier, these
Can
various
you
proteins,
specify that, because that appears to be what his entire argument hinges on, that there is no one, that there's no, without appealing to the theory, without hypothesizing, I guess is what he's saying, that we don't have a laboratory experiment that verifies that these novel protein folds can be stable and produced evolutionarily,
but we have
and that
all
there's
these,
these bridges. I'm sorry, go ahead.
yeah, we've got all these different subsets, and they're all stable and functional, and they do useful and interesting things for us. So that claim falls apart pretty quickly. But then also when we look at these particular proteins, that for instance, look at a transporter protein. A transporter protein is typically a transmembrane molecule, so it crosses the membrane. It allows select molecules to be shuttled from the outside to the inside, or vice versa. And when we look at those, what we find is that it's not a passive thing. There are active transporters that contain kinases and proteases. So there's an easy route to transform a transporter into an enzyme that's functional in modern cells. You just got to separate out the different components. It's like there are various cellular markers where we can actually take them apart molecularly. We can see that, okay, this protein has a transmembrane domain, it has an intracellular domain that interacts with certain proteins, and it's got an extracellular domain that is a recognition site for some other protein. And when we look at across the whole group of animals, we can find cases where bits and pieces of it have been swapped out and exchanged with each other. So we can see these proteins in a family. Um, it's like those kids' books where you've got the 3 pages you can flip. There's the head, there's the body, and the legs, and we can look at these proteins. We can say, "Oh, yeah, this one's got that central part, but it's flipped to this other extracellular domain." So it's not like that's impossible or not observed. We see that all the time when we look at these protein sequences.
So then, one of the other things that I said, what the guy refuses to admit what evolution is, right? So you don't teach a high school class, you teach college class. He
Right.
said evolution is not really varying allele frequencies. That works for high school kids, but it falls short. So you, as a college professor, is that an accurate definition of evolution, or does it fall short?
Oh, you're putting me on the spot here because I
Well,
will tell
when I
you,
say does it fall short, I'm
yeah, as a developmental biologist, that's not a complete story, but it is the fundamental story behind evolution.
well, you can't
Uh,
say that it's not really varying allele
Correct.
frequencies, can you?
And I have taught evolutionary biology at the college level, and yeah, I teach them about it's about changes in allele frequency over time.
What I said about the different families, or the different, when you look at Canidae, for example, and you look at the different groups of foxes that cannot interbreed with each other, the different species of dogs that cannot interbreed with each other, and then of course then the some groups like the wolves, coyotes, and domestic dogs that can interbreed with each other. Those are all an evolutionary relationship. What I would say, and
Mhm.
can you tell me whether I'm right or possibly flawed in describing that when you have the genome, one ancestral genome producing two genetically distinct subsets, is that not evolution by definition?
That is, yeah, that's evolution.
Okay, so if you accept that all dogs are related, then you must accept that evolution is a fact.
I would say so.
Yeah.
Apparently, this guy doesn't.
Yeah, he wants to call it adaptation without knowing what he
Oh,
doesn't know what words mean.
yeah, he's reciting Discovery Institute nonsense, isn't he?
Of course.
Yeah, yeah.
Somebody said today that something that the Discovery Institute was coming out with was very compelling, and so I asked him. I haven't got a response yet. I'm like, tell me something that the Discovery Institute came out with that is compelling, and I'll make a video about it if I haven't already refuted it.
Yeah, I haven't looked in on evolution news from the Discovery Institute for a week or 2. Uh, I saw nothing then. I don't Do you think maybe they've come up with something radical and exciting in the last couple of days?
I just want to clarify, but I don't want to go over too much. He says that there's no possibility confirmed, like in laboratory experiments, for producing new, stable, novel protein folds within the evolutionary. Do we have confirmation that this is in fact possible?
Yes, we have
Okay.
confirmation that that happens routinely.
So there
Yeah.
is research published that explains how a new protein family can evolve even in the rugged evolutionary landscape.
Okay. Again, the rugged evolutionary landscape is it's an old idea. I don't particularly like it, but yeah, that we can evolve all kinds of interesting things that have been observed. So I mentioned, transport proteins. Transport proteins have been studied pretty extensively, and when they examine transport proteins, what they see is mundane, trivial changes, intragenic modules that get repeated to build a more complex structure, uh, that are all entirely feasible with simple genetic mechanisms.
Now, you mentioned in a private correspondence earlier, you mentioned gene duplication as being the ultimate explanation for this. And I know that this guy has already tried to, he said gene duplication isn't good enough. Kind of like a Michael Behe, this isn't good enough.
Yeah.
Would you mind explaining how gene duplication really does apply here, if in fact it does?
Well, I sent you something. I sent you
I can
this.
read it. I mean, I got it in front of me. You said that in eukaryote genomes, there are many kinds of gene families. Gene duplication and conversion are sources of the evolution of gene families, including those with uniform members and those with diverse functions. Population genetics theory on identity coefficients among gene members of a gene family shows that the balance between diversification by mutation and homogenization by unequal crossing over and gene conversion is important. Also, evolution of new functions is due to gene duplication, followed by differentiation. Positive selection is necessary for the evolution of novel functions. However, many examples of current gene families suggest that both drift and selection are at work in their evolution. Now, this guy has already said that all of that doesn't apply. And he doesn't give a reason why. He just says that none of it applies or that none of it is
Yeah,
good.
so he's smarter than Tomoko Oda. Yeah,
Yes.
right.
Because, all of these intelligent design people are have no better about subjects they've never studied than all the best educated expert specialists anywhere ever.
Yeah. Well, one of the key phrases in that abstract that I sent you was that Oda was mentioning how unequal crossing over is a mechanism. Uh, Unequal crossing over is a really easy mechanism to understand that, during meiosis and all that stuff, you are duplicating genes, you're duplicating chromosomes, and they line up, they pair up, and there's crossing over events that can exchange bits and pieces of the chromosome. Um, Normally, if everything's lined up perfectly, you just get a duplicate of the chromosome. But if they're off a little bit, if they're just offset just a little bit, they're not perfectly aligned, and you get a crossing over event, it can lead to a deletion or duplication of a big chunk of the chromosome. So gene duplication is trivial, happens all the time. That we can look within humans even, and we can see examples of gene duplication. There's copy number variants occur in different groups of people at different rates and different frequencies, and we can measure that. So we know that these duplication events happen all the time, routinely, and they yield functioning individuals. This doesn't disrupt the development or function of the organism at all. So, when people tell me that, oh, okay, that's not possible, gene duplication can't play a role, I just have to tell them, if I could sequence your genome and compare it to mine, I would probably find various gene duplications all over the place in our genomes. It just happens. And then, of course, the next step is, if you've got a duplication of a gene, you can go on and diverge. You can accumulate mutations in one copy or the other, and leave a functioning copy and leave a copy that has a new function. It's pretty easy to imagine how that happens. And when I say imagine, I don't mean I'm inventing it. I mean, we can go into different organisms, sequence regions of their genome, and see it right there that there has been a gene duplication event.
He says that without discovery of some sort of bridge, or this particular bridge in the gap that he's imagining here, he says that protein evolution is inconceivable, and he says that no bridge has been found. Can you elaborate on how there
Sure.
has
Yeah.
been?
Well, what we do have is we have examples of gene duplications in different related organisms where some of the genes have gone off in other directions. Um, One of the examples I use in my introductory biology course is the antifreeze gene in various fish. And what you find there is there was a gene duplication 40 million years ago. We can see the duplication. It's also very nice because there were multiple gene duplications, so we've got intermediate stages of this preserved as non-functional DNA in the genome. And we can see that one copy retained its function as a pancreatic enzyme. Can't get away without that. You need that to start digesting your food. And one copy got duplicated, then got truncated into a simple trinucleotide repeat that functions as an antifreeze protein. But we can see the structures. We can see, the the regulatory regions. We can see all the adjoining DNA, and we can see that those two genes have a common origin in a duplication event. So, that's the evidence, is that we can actually see it in the genome when you look closely enough. So yeah, that's the bridge. Is in this case, gene duplication was the bridge that produced an additional copy that could function in the organism.
Thank you. Because I think his ultimate point is in this one sentence where he said that there is no evidence that evolution is possible. And he said this after I gave him genetic confirmation that evolution had happened, at least within dogs. Because
Right.
that was, of course, the thing that's the thing that all of these people bring up, is that it's in dogs. And I remember what I wanted to say before. That they all of these intelligent design arguments, they seem to accept that evolution happens. It seems to be that most of the ID proponents actually accept that evolution does in fact happen, but their argument is always with either the origin of life or, more interestingly, with the origin of eukaryotes. And no issue with evolution beyond that. And that's where
Right.
this guy's coming from, I think, with his going back to the Cambrian explosion.
Yeah, well, eukaryotes, that was what, 3 billion years ago? I would be so happy if the creationists would just come out and admit that the last 3 billion years of evolution happened, and they happened by the mechanisms that we have proposed.
All right,
And
so
then
let me
we can
go
work
back
on
over
that
this.
3 billion year event.
Let me go back over his initial point then one more time, just to clarify. We'll end it here. He says there is no evidence that evolution is possible, despite all of the proof that evolution has happened. There's no evidence, and the mechanisms by which it happened that have been confirmed. He still says there's no evidence, which is a bald face, out and out lie. All of these facts in evidence that evolution is possible. This is how it happens. This is when it happens. This is where it happens. This is, we see all of it. He says, and none of it is possible because protein evolution has no known solution. And your final statement there?
I don't even know what he means by no known solution. That that yeah, that's the key thing, that when we talk about, maybe you're used to talking about like evolution of dogs and macroevolution, all that kind of thing. Uh, but in my experience, most evolutionary biologists are more interested in the molecular mechanisms. And there we say, yeah, it's really clear that there are simple molecular mechanisms that can produce the variety of proteins that we see in various organisms.
Outstanding. PC Myers, thank you very much.
Anytime.
I hope you'd appreciate this other softball. Thank you very much again.
Yeah, it's that was pretty soft and gooey and easy to deal with. I doubt that he will accept anything I said, but that's okay.
Oh yeah, I have no doubt. When I showed him proof of what evolution is and what he had already admitted to, his best answer is no.
Yeah, it's impossible,
Oh, he said,
inconceivable.
I asked him if there are any facts. He says, how can we consider evolution a fact if there are so many facts against it? I said, what are the facts against it? And he said, free will and morality. Like, here's
Oh
somebody that
boy.
doesn't know anything.
Okay.
Here's somebody who doesn't know dick about Jack.
Yeah. Oh yeah. Well, we can't talk about genes that regulate morality, so I guess we just have to throw up our hands on that one.
Do we have genes that regulate free will?
If free will even exists, I have my doubts.
Exactly. And can it exist in a realm where God can decide to harden Pharaoh's heart and decide to play both sides of the chessboard that way?
Right. Yes.
Thank you very much, sir.
Thank you. Like I say, anytime.